All right, let's jump right in. We are going back in time, all the way to 1929, to a paper in the British Journal of Experimental Pathology. And at first glance, it doesn't look like much. But this single document by a bacteriologist named Alexander Fleming is what kicked off the entire antibiotic revolution. This is the story of that paper.
So it all starts right here, with what you might call a happy accident. Fleming was working with staphylococcus, a really nasty bacteria, and he noticed something strange on a culture plate he'd forgotten about. There was this blob of mold growing on it. But the crazy part, the part you can see right here, is what's happening around the mold. The bacteria, they're just dissolving. They're becoming these transparent ghosts. Fleming called it lysis. He knew, right then and there, he was looking at something big.
You know, that one observation just opened up this huge question. What in the world was this mold making that could just wipe out bacteria like that? And more importantly, was this just a weird fluke in the lab, or could this invisible weapon actually be controlled, harnessed, and used for something? This is the scientific detective story Fleming set out to solve. Okay, so picture it, Fleming's lab at St. Mary's Hospital in London. You've got culture plates everywhere, and stuff from the air is always falling into them. Contamination is just part of the job. But instead of just tossing this plate in the trash, Fleming took a closer look. And that right there, that single moment of curiosity, is what separates a ruined experiment from a Nobel Prize. So he identifies the intruder, and get this, it wasn't some exotic, rare microbe from a faraway land. It was a species of Penicillium, a super common mold, the kind of thing you might find on old bread. He even noted that this type of mold was, quote, not uncommon in the air of the laboratory. The miracle killer was literally floating around them the whole time. Just incredible. All right, so Fleming has his mystery mold. Now what? Well, he started growing more of it in a broth and began this really systematic process to figure out what exactly this bacteria-killing substance was. He had to isolate it, test it, understand its properties, and of course, he had to give it a name. And where did that world-famous name come from? You know, it wasn't anything grand or poetic. It was pure practicality. As he wrote himself, he just wanted to avoid the cumbersome phrase, mold broth filtrate. So he just shortened it to penicillin. It's a simple, efficient label for his mold juice. So Fleming starts building a profile, right? He's running tests. First question, can you even work with this stuff? Turns out, yeah, it dissolves in water. You can pass it through a superfine filter. Pretty easy to separate. Okay, but is it tough? And this is where it gets really interesting. He discovered it could even survive being boiled for a little while. He was methodically putting the pieces of this puzzle together. Now we get to what might be the single most important discovery in this entire paper. This was a total paradigm shift. See, the disinfectants they had back then were basically just brutal poisons. They killed everything they touched. But penicillin? No. This was something far more elegant, way more subtle, and ultimately so much more powerful. It was a selective killer. And you can see it perfectly in this brilliant, simple experiment. He puts a strip of penicillin down the middle of a plate and then streaks different bacteria towards it, kind of like he's daring them to cross the line. And look at that. Some bacteria just stop dead in their tracks. There's this invisible wall they can't cross. But others? They grow right up to the penicillin, completely unharmed. And here's the data, basically penicillin's hit list. On the left, you have all the bacteria it completely wrecks, the major players in wound infections, pneumonia, strep throat. But on the right, you have the ones it doesn't touch at all, like the germs that cause typhoid. The point is, this wasn't a bomb. It was a sniper rifle. This is the very beginning of a whole new way of thinking about medicine. So, just how powerful was this stuff? Fleming found that a good batch of his mold broth could completely stop staph from growing, even when it was diluted 800 times. Just let that sink in for a second. 1 part penicillin in 800 parts of liquid was enough to shut down one of our deadliest bacterial enemies. That is some serious potency. Okay, this is where the story takes a really fascinating, unexpected twist. You've just discovered this amazing bacterial killer. The first thing you'd think is, let's treat patients, right? But that's not what happened. Fleming's paper reveals that his first, most immediate use for penicillin was actually as a tool for his fellow scientists. So here was this common problem bacteriologists had. Sometimes they needed to study a really picky, slow-growing microbe, like bee influenza. But when they took a sample, more aggressive bacteria like staph would just grow like crazy all over the plate, completely hiding the one bug they were actually looking for. And penicillin was the perfect solution. I mean, look at this. On the left, you've got this total mess of overgrown bacteria. But on the right, Fleming adds some penicillin. It's like a selective weed killer. It completely wipes out the staph weeds, but it leaves the penicillin-resistant bacteria behind, allowing it to grow into a nice, pure culture. It's absolutely brilliant. And in his summary, this is what Fleming really emphasizes. Not a miracle cure for humanity, not yet, anyway, but a practical lab technique. He says flat out that its value as a tool has been demonstrated. This was the first proven concrete application for penicillin. But Fleming knew. He knew he was sitting on something so much bigger. And as you get to the conclusion of his paper, you can almost feel the sense of possibility growing. Because he'd done one more critical set of tests that hinted at penicillin's true world-changing potential. This slide right here lays out the absolute game changer. The antiseptics they had back then, like carbolic acid, were just brutal. They operated on a scorched earth policy. Sure, they killed bacteria, but they also killed the patient's own cells. They were toxic. But Fleming found that penicillin was completely different. It was lethal to bacteria, but it was remarkably non-toxic to animal cells. This was the holy grail, a substance that could kill the invader without harming the host. And that incredible discovery led him to write this sentence. It is so understated, but it might be one of the most revolutionary statements in the history of medicine. He suggests it may be an efficient antiseptic for injection into infected areas. That's it, right there. That is the birth of the idea of an antibiotic, a medicine that could fight infection from inside the human body. So Fleming laid it all out in this one incredible paper. The accidental discovery, the selective power, the non-toxicity, and the unbelievable therapeutic potential. He had lit the fuse. The challenge now, the big question left hanging, was how to turn this mold broth filtrate into a miracle drug. Fleming had started the race, but now it was up to other scientists to pick up that torch and carry it over the finish line.